Welcome to PMART. This video will describe the file requirements when uploading personal data files for analysis. A written version of the file requirements is available on the PMART website once you select the Upload Personal Dataset experience. Additional tutorial videos on uploading personal datasets and performing an analysis are available on our YouTube channel. Analyzing personal datasets in PMART requires 2 or 3 CSV files to be uploaded to the website. From the PMART homepage, select the Upload Personal Dataset Files experience. From here, you will be asked to upload your files. Now I'll show you some example files. This is the expression data file. This particular file gives peptide information and contains quantified data for each biomolecule, the rows, and each sample, the columns. The first row in this file is the header and contains names for each column. One of the columns must contain the unique biomolecule identifiers. In our case, we are uploading peptide data, and the name of our identifier column is peptide, column A. All remaining columns in this file contain the quantified values for each sample in the dataset. The column names are the sample identifiers. In this case, they are numeric. In the expression data file, missing values can be denoted with one of the following: NA, as seen here, NaN, a blank, or a 0. The data can be unnormalized or already normalized, and it can be on one of the following scales: raw, log 2, log 10, or natural log. Second required file is the sample information file. This file contains information about each sample, for instance, clinical or other variables of interest. Again, the first row is the header and contains the names of each column. One of the columns in the sample information file must contain the sample identifiers, and these identifiers must be exactly the same as the sample identifiers that are the column names in the expression data file. Every sample in the expression data file must also be contained in the sample information file. The remaining columns in the sample information file contain clinical variables of interest or other sample-specific information that may be relevant to the dataset. In our example file, we have information on the patient's race, diagnosis method, vital status, and others. Information to be used for making statistical comparisons should be contained in the sample information file. The third file is optional, and this is a metadata information file. This file contains a mapping of the biomolecule listed in the expression data to other types of biomolecules. For instance, a mapping of peptides to either proteins or genes. In this file, the first row is the header and contains the names for each column. There must be a column containing the same unique biomolecule identifiers as found in the expression data. The column name must match and the biomolecule values must match. For example, in this file, column G is titled peptide and contains the peptide information. This corresponds to column A in the expression data file, also titled peptide. It's important to note that each peptide found in the expression data file must have at least one corresponding entry or row in the metadata file. There should be at least one additional column in the metadata information file that contains a mapping of the expression data biomolecule identifier to another identifier. In our case, there is a column for peptide, and there are also columns for protein and gene that give the proteins or genes that each peptide maps to. If quantitation from peptides to either proteins or genes is desired, the metadata information file must be uploaded and contain a mapping from peptides to either proteins or genes. The mapping in the metadata information file is also used in the Trelliscope displays to link one identifier to another. For instance, if protein information is included, the Trelliscope displays will include a link to the UniProt webpage for the associated protein. and similarly a link to the GeneCards webpage for the associated gene if gene information is included. A separate video is available on the PMART YouTube channel to show how to upload your own files to PMART.